Screen. Projector. So, hello. So in this session, we are going to talk about more of the on neurons, microscopic structure of the neurons. We are going to talk about synapses, and we are also going to talk about the glial cells, the different types of glial cells, how they look like, and what is their function. This is not actually, as stated, the histology of the nervous system, because histology of the nervous system, we are going to deal with the peripheral nerves, we are going to deal with the cerebellum and cerebrum, and that would be done mainly in the labs. There would be 1 or 2 labs where you would have a complete section where we are going to deal with the histology of the nervous system. But in this session, we are going to talk about more on the neurons, the classification of neurons based on their structure, shape, and other things, and their function. We are going to talk about the different types of glial cells and what is their function. We are going to talk about synapses and how the synapses work. So those type of things which are going to be the core of this session. And then we are going to talk about a little bit on some of the diseases related to these aspects which we are talking today. So if we look at a neuron, we would find that it is a type of cell, but it's a little bit different type of cell which we see. You would find a cell body, and then there are multiple processes which are called neurites. Now, if you see the processes which are coming out from the cell body, these are called the dendrites. And dendrites receive synapses or neurites from other neurons, and that's why you can see on the top of the dendrites there are dendritic spines where there is synapse from axons of the other neurons. Then there is a long neurite which is coming out from the cell body, and this long neurite is called the axon. It starts at a place which is called the axon hillock, and most of this long neurite, which is the axon, is covered by myelin sheaths. And these myelin sheaths in the central nervous system, as we said, they are formed by the oligodendrocytes, and in the peripheral nervous system, they are formed by the Schwann cells. Schwann cells wrap around these axons to form the myelin sheath. The purpose of these myelin sheaths is that they provide insulation. So once the electrical activity which is generated at the axon hillock it passes from one place to the other, uh, and it is insulated by the myelin sheath. So these are nodes of Ranvier where you have spaces which are not covered by myelin sheath. So the electrical activity which starts at the axon hillock passes from here to the nodes of Ranvier, and then it keeps on jumping till it reaches the axon terminal. And then once it reaches the axon terminal, you can see that axon terminal is also divided into button-shaped dilatation. These are called axonic buttons, which are basically synapsing or joining with either target organs, which could be another neuron, which could be a muscle, which could be an organ. So you can have the axons synapsing or dividing into the axon terminals, which are dilated to form axon buttons, and they are going to synapse with either the muscle or another neuron, or they are going to synapse with any other organ. If you look at the cell body, it has a nucleus, and then what you typically see is a granular cytoplasm. So these granules which you see in the cytoplasm of a neuron is basically called Nissl bodies. And that's how you identify neurons, because you see in the cytoplasm granular appearance, and that granular appearance is because of Nissl bodies. And the Nissl bodies are stacks of rough endoplasmic reticulum. So they are distributed all around the cytoplasm, giving the granular appearance to the cytoplasm. They are only not present at the axon hillock. So most of the neurotransmitters are proteins. So you can imagine that they are going to be synthesized in the cytoplasm through the rough endoplasmic reticulum, and then they would be packaged in the Golgi apparatus, and then transported through the axon to the axon terminal, where they are going to be stored until the impulse comes in, and then the package is released into the synaptic cleft. Okay. There are four functions of neuron. So they receive and integrate inputs from the different other neurons, and that is basically done in the cell body of the neurons. So dendrites receive it. The cell body of the neurons are going to integrate these inputs. Then they would generate an action potential, which is going to be generated at axon hillock, transmitted this action potential through the axon, through nodes of Ranvier. They would keep on going down, and then transmit the information to the target cell through the synapse with another neuron or a muscle at the neuromuscular junction, or a gland where the secretomotor fibers are there. So this is done at the axon terminals or nerve terminals. Now you can see it here. The dendrites are receiving the information, and this is basically integrated in the cell body. Then the overall effect on the cell body, both from the excitatory and inhibitory neurons, is going to result in a generation of the action potential, which is going to start at the axon hillock, and then will move down and will stimulate other neurons. So this is the normal way how the nerve impulses are basically generated. Now, this is a typical neuromuscular, or a synaptic terminal. So what you are basically finding is that the axon terminals are dilated to form axonic buttons, and these axonic buttons have vesicles which contain neurotransmitters, and they also contain these organelles which are mitochondria, which will generate energy for these neurotransmitters to be released into the synaptic cleft. Once the neurotransmitters are released in the synaptic cleft, they find receptors on the postsynaptic dendrites, where they are going to bind and result in opening of either the sodium channels or opening of the potassium channels, depending upon what type of neurotransmitter is binding. If it is an excitatory neurotransmitter, it is going to bind to the receptors, resulting in opening of the sodium channels and resulting in action potential to be generated. On the other hand, if it is an inhibitory neurotransmitter, it is going to bind there, resulting in opening of potassium and chloride channel, resulting in inhibition of the action potential. So in terms of classification of neurons, we can classify neurons based on their functions. For example, they could be sensory neurons, they can be motor neurons, they can be interneurons, which are joining the sensory and motor neurons. So if you look at the example of a sensory neuron, the most important example of a sensory neuron is the dorsal root ganglia. And if you look at these neurons in terms of their shape, the dorsal root ganglia neurons, they are quite convoluted type of neurons. Their axons are quite convoluted. They have a big cell body, then the axon is convoluted, and then it divides into this one single axon, which is coming out, is going to divide into two processes. One is the central process, the other is the peripheral process. Peripheral process is going towards the receptors in the skin to take the sensory input, and the central process is going into the spinal cord to relay into the dorsal gray horn cells, where the sensory neurons are present in the spinal cord. So one of the example of the sensory neuron is the dorsal root ganglia. And if you look at this neuron, it is much more convoluted. It has a big cell body, and the neurite which is coming up is convoluted. It divides into a central process and into a peripheral process. An example of a motor neuron, on the other hand, has it is basically present in the ventral gray horn cells of the spinal cord, and it has several processes coming out. So it is a multipolar type of neuron. Multiple neurites are coming out. So it has multiple dendrites and one single axon. You can see it here also in the electron microscopic structure. You can see multiple, dendrites are basically there. So you can see this, and there is also with these motor neurons, there is multiple synapses happening there. So you can see the dendritic spines which are there, where there is multiple axons synapsing with one motor neuron. Then you can see the nucleus, and you see this granular appearance. This granular appearance is the Nissl body, which is basically the stacks of rough endoplasmic reticulum. The other classification of neurons can be based on their structure. So we did the classification based on sensory, motor, or interneurons. We can also classify neurons based on their structure. It could be unipolar, bipolar, and multipolar. So you can have a unipolar neuron with one neurite coming out from the cell body. You can have a bipolar neuron which has two neurites coming out, and you can have a multipolar neuron which has multiple neurites coming out from the cell body. So this is the example of unipolar, this is bipolar, and this is multipolar. Now, the classical unipolar with only one neurite coming out usually does not exist in vertebrates. It's mainly you would find this classical unipolar type of neuron in invertebrates. The type of unipolar neuron which you would find in humans and mammalian in mammals is this type of unipolar neuron, which is a pseudo unipolar neuron. So you have one neurite coming out, but it is dividing into two processes, one going towards the periphery, one going towards the central process, which is going towards the spinal cord. This is the type of neuron which you would see mostly in the dorsal root ganglia. So the dorsal root ganglia has the pseudo unipolar type of neuron. It's not the classical unipolar where there's only one neurite. This is usually present in invertebrates. We can have a bipolar neuron, which has two processes, one the dendrite, the other is the axon. This you can see in the retina. The bipolar cells of the retina is an example of a bipolar type of neuron. Then you have the multipolar neurons. Multipolar neurons, you can see it at many places. One of the examples we have discussed is the anterior gray horn cells or the ventral gray horn cells of the spinal cord, where you would have multiple dendrites and one axon. The other example is the pyramidal cells of the cerebrum and hippocampus, where you would see the cell body, which is triangular in shape or pyramidal in shape. That's why it is called pyramidal cells. And then you have the dendrites, and you have one axonal process. The other type of multipolar cell is like a tree. You can see the dendrites here, and this is the Purkinje cell of the cerebellum. So this is also an example of a multipolar type of neuron. Purkinje cell of the cerebellum, pyramidal cells of the cerebrum, and the motor neurons of the anterior gray horn cells of the spinal cord. They are all examples of multipolar type of neurons. You will also hear, based on the length of the axon, two types of cells, which are Golgi type 1 and Golgi type 2. Golgi type 1 has long axons. They can be up to 1 meter long. And Golgi type 2 has small, axons. So based on the length of the axon, also you can divide the neurons into Golgi type 1 and Golgi type 2. Golgi type 1 has very long axons, and Golgi type 2 are smaller neurons with smaller axons. Then based on the shape, you can give them the name pyramidal base because you saw the cell body triangular, so you gave them the name as pyramidal. Stellate, if they have a star-shaped cell body, then they are called stellate cells. If they have a more of a basket-shaped cell body, then it's basket cells, granule cells. So based on the shape of the cell body, the neurons also have been given their names. Pyramidal cells in the cerebrum, stellate, basket, and granule cells you would find in the cerebellum. So neurons are also classified based on the shape of the cell body. So these are the different shapes you can see. So based on the different shapes of the cell body, the neurons have been given different names. Then we come up with the effect of the neuron. It could be an excitatory neuron, or it could be an inhibitory neuron. So based on the neurotransmitters which they are going to release, they can be excitatory. For example, in the central nervous system, if a neuron is releasing glutamate, it is an excitatory neurotransmitter, and the neuron would be an excitatory neuron. If it is releasing GABA, for example, it is an inhibitory neuron. So because it is releasing a neurotransmitter, which is an inhibitory neurotransmitter. Similar is the case in the peripheral nervous system. You have acetylcholine, which is an excitatory neurotransmitter in most places. So you can divide the neurons also based on their the type of neurotransmitter and the effect of the neuron. You can divide them into excitatory and inhibitory neurons. So we can divide them based on function, sensory, motor, interneuron, based on their structure, unipolar, multipolar, pseudo-unipolar, bipolar, based on their shape, where in different types of cell bodies which are present in particular shape of neurons are present in particular parts of the brain. For example, pyramidal neurons are present in the cerebrum. Purkinje type of neurons or granule cells or basket cells are present in the cerebellum. So based on the shape, you can classify them, and then based on the effect of the neurons can be classified. Now we are coming to the types of the glial cells. So we have talked about neurons. The other type of cells which you find in the nervous system are the glial cells, which are the supporting cells of the nervous system. So the first one are astrocytes. So there are two types of astrocytes which you find. Astrocytes because they look like star-shaped type of structures. That's why they are called astrocytes. And there are two types of astrocytes. One are fibular, fibular, fibular astrocytes, which are present in white matter mainly. And the other one are protoplasmic astrocytes, which are present in the gray matter, or where you have the cell bodies of the neurons present. So you have two types of astrocytes. One are fibular and the other are the protoplasmic astrocytes. Now there are multiple functions of the astrocytes. One of the functions of the astrocytes is that it insulates the synapses. These astrocytes, if you see, they have uh, you know uh, processes, several processes, and their processes has foot processes which cover the capillaries, the gap junctions of the capillaries. So because they cover the capillaries, the astrocytes in the central nervous system, they are also going to form the blood-brain barrier because they are going to insulate the capillaries. Their processes are attached to the surface of the capillaries, so they are going to form the blood-brain barrier. They are going to insulate the synapses. They have a function of, phagocytosis of the debris in the of the axons. They have the function to maintain the metabolic function in the microenvironment around the neurons. Their food processes also form outer limiting membrane and the inner limiting membrane. That means the food processes are attached to the inner surface where the ventricular cavity is, and on the outer surface where the pia mater is. So on the outer surface of the brain, you have the pia mater. So their food processes attached to the inner aspect of the pia mater, and their food processes are also attached to the inner part of the ventricular cavity. So they are they are forming the inner and outer limiting membranes. Uh, Astrocytes are going to, wherever you have the injury in the brain, if the neurons are not regenerated, the space is going to be occupied by, or the debris is going to be cleared up by the astrocyte, and the process is called gliosis. So you would have gliosis or fibrosis like what you have in the other places. You have fibrosis in the brain. Wherever you have neuronal damage, you would have proliferation of the astrocyte, and the process is called gliosis. If you have a tumor coming out from the astrocytes, it is called glioblastoma, which is one of the malignant tumors of the glioma or glioblastoma. These are malignant tumors of the brain. The neuronal tumors, the neuroblastomas, are only present in children and they are malignant. But the glioblastomas and gliomas are the ones which you would find in adults, and they also come out from the astrocytes. The other type of glial cells are microglia, which are small star-shaped type of cells. They do not originate, as we talked about in the embryological session also. These are the only glial cells which come out from the mesenchyme, and they are the antigen presenting cells, or the resting macrophages within the central nervous system. So whenever you have uh, an infection, they would act as antigen presenting cells for immune cells and the immune reaction. Oligodendroglaya, they are the ones which are important for the myelination of the neurons in the central nervous system. And one of the diseases which results in demyelination of neurons in the central nervous system is multiple sclerosis. So if you have heard about MS, it's a demyelinating disease. So the antibodies are produced against the myelin sheath in the central nervous system, resulting in demyelination. So oligodendroglaya are important in terms of myelination of neurons in the central nervous system. Schwann cells, on the other hand, are important for the myelination of neurons in the peripheral nervous system. So if you look at these, you would find there is protoplasmic astrocytes, and these are the fibrous astrocytes. You see the foot processes of these astrocytes are covering the capillaries in the central nervous system, forming a blood-brain barrier. You can also see the processes of these astrocytes attached to the inner layer of the ventricular cavity and also to the inner layer of the pia mater, forming the outer glial limiting membrane and the inner glial limiting membrane. They are also attached to the neurons and to the axonal processes of the axon, so that they can insulate certain neurons, certain neurotransmitters which you don't want to affect the other neurons nearby. So the food processes of the astrocytes are going to insulate the axon terminal. They are also important in terms of the metabolic exchange between the neurons in the microenvironment in the central nervous system. These are the microglia, which you can see very small cells with multiple processes. These are the phagocytes and the antigen presenting cells of the central nervous system. Then you have the oligodendroglaya, which are forming the myelin sheaths over the axons in the central nervous system. And one of the diseases which you should remember in terms of demyelination is the multiple sclerosis. So the function of the astrocyte is mechanical support. It forms a framework, and in embryological life, these astrocytes are providing a framework through which the neurons are going to migrate to different places. So it's mechanical support, and it's a framework which is the astrocytes are providing, through which in embryological life, the neurons are basically migrating using the scaffold of the astrocytes to reach at different parts of the brain. It provides a metabolic support, transport of nutrients and waste, encapsulate synapses so that it does not affect the nearby neurons, regulate the chemical and ionic environment of the central nervous system, forms the scar tissue after the death of the neurons. So you have gliosis there, act as phagocytes to take out the debris of the dead neurons. This is the picture of the astrocytes. You can see there's star shape, and that's why they are called astrocytes. Microglia, we know that they act as resident macrophages of the central nervous system. They act as phagocytes, and they act as antigen presenting cells for the to the immune system in the brain. This is the shape of the microglia. You can see them here. Oligodendroglaya, they are important in terms of myelination of the axons of the central nervous system. You can see that each oligodendroglaya has multiple processes, and these multiple processes are myelinating different axons, different parts of axons of different neurons. If you compare this with Schwann cells are going to myelinate, each Schwann cell is going to myelinate one axon. So you see here, Schwann cell is wrapping around one axon, while you would see the oligodendroglaya has multiple processes. So each process is going out and myelinating a part of the axon of different neurons. So here, In terms of the peripheral nervous system, where the Schwann cells are myelinating, you have one Schwann cell wrapping around one axon. While in terms of the oligodendroglaya, it has multiple processes, and each process is myelinating different components of the axons of uh, different neurons. So you can see it. This is the myelination process. You can see one oligodendroglaya has several processes, and it is myelinating different axons of different neurons. While you would see Schwann cell is wrapping around one axon terminal. So this is the difference. One difference is that it is the oligodendroglaya are important in terms of the peripheral nervous system, the central nervous system, myelination of the neurons in the central nervous system, while Schwann cells are myelinating the nerves in the peripheral nervous system. The other one is that because oligodendroglaya has multiple processes, and each process is basically myelinating different axons of different neurons, while one Schwann cell is going to wrap around one axon and basically myelinating one part of the axon. Now we are coming to the synapses. There are two types of synapses in the nervous system. The most common one is the chemical chemical synapses. Synapse means where one axon of the neuron is going to join with uh, the cell body or the dendrite or the axon of the other neuron. So the space between them or the joining of one neuron to the other neuron is called a synapse. Now this synapse uh, could be chemical. So there is a release of neurotransmitter, and this neurotransmitter binds to the receptor, resulting in an ionic change, which result in excitation or generation of action potential. That is called a chemical synapse. There could be, there are electrical synapses in the central nervous system also, which means that one neuron is joined to the other neuron by gap junctions. And there is ionic exchange between one neuron to the other through gap junctions. And those are called electrical synapses. In electrical synapses, you have bidirectional flow. So the action potential can move this way or the other way because there is gap junction and there is exchange of ions between the neurons. But there are very few electrical synapses. The most common way of how the neurons communicate with the other neurons is by chemical uh, synapses. Now, what happens in chemical synapses is that you have these neurotransmitters which are present in the axon terminal. And when there is binding of the calcium to the terminal part of the axon terminal, this results, or the calcium influx into the axon terminal, this makes these vesicles, which are containing neurotransmitter, to bind to the axon terminals and release their payload. Once these neurotransmitters are released, they are going to bind to the receptors on the postsynaptic membrane. When they bind to the postsynaptic membrane, these receptors, the neurotransmitters bind to them. If they are excitatory neurotransmitters, this will result in opening of the sodium channels, which will result in the generation of action potential in the postsynaptic membrane. On the other hand, if the neurotransmitter is an inhibitory neurotransmitter and binds to the receptor here, it can result in opening of the potassium and chloride channel, which result in further negative hyperpolarization of the postsynaptic membrane and inhibition of the action potential. So the first event which happens is the calcium influx, which results in the vesicles which are containing the neurotransmitters to bind to the synaptic membrane and release their payload. These neurotransmitters, once they are released in the synaptic cleft, they go and bind to the receptors on the postsynaptic membrane, resulting in either opening of the sodium channel and excitatory postsynaptic potential, or opening of potassium and chloride channel, which results in inhibitory postsynaptic potential. Now, there are two, you know, syndromes which you should remember based on the neuromuscular physiology. I'm not talking about the postsynaptic membrane as such, but in terms of the neuromuscular junctions where a neuron is binding to the muscle and releasing acetylcholine, you should remember at least two of these syndrome. One is the myasthenia gravis, where you have the antibodies which are similar to um, you know the acetylcholinesterase. Acetylcholine receptors. So they can go and bind to the acetylcholine receptors, resulting in continuous sodium influx and excitation. And as a result, there is, over the time, there's fatigue in the muscles because you have these antibodies which have a similar shape to acetylcholine, and they go and bind to the acetylcholine receptors on the neuromuscular junction, and they keep on stimulating the sodium channels to remain open, resulting in continuous contraction of the muscle. And ultimately, there is fatigue which happens. The other syndrome which you should remember is the Eaton-Lambert syndrome, because Eaton-Lambert syndrome would also, present with muscle fatigue. But what happens in Eaton-Lambert syndrome is that the antibodies are against these calcium, channels. So what happens is that there is a continuous influx of the calcium, and as a result, these neurotransmitters are continuously released. So in Eaton-Lambert syndrome, there is antibodies against the autoantibodies are against the calcium receptors, while in the myasthenia gravis, you have the antibodies for the acetylcholine receptors. But both of them are going to present in a similar fashion. Excitatory postsynaptic potential, we already talked about that binding of the neurotransmitters to the postsynaptic receptors result in opening of the sodium channels to diffuse into and potassium to diffuse out, resulting in excitation and generation of the action potential. Inhibitory postsynaptic potential happens when you have an inhibitory neurotransmitter binding to the postsynaptic receptors, resulting in opening of either potassium to diffuse out or chloride to diffuse in. This is going to result in inhibitory postsynaptic potential. Now we have EPSP and IPSPs together. So if the excitation is subthreshold, then there would be no excitation. If there are two excitations coming at the same time, there is temporal summation. If there is spatial summation, it's when the excitation is coming to the same neuron but at different places. So that is spatial type of summation. You can have excitation and inhibition coming together, and if they cancel out each other, then there would be no excitation of the neurons. So these are the different types of scenarios which you can find based on the excitation, postsynaptic potential, and inhibitory postsynaptic potentials. Synapses, in terms of the anatomical synapses, you can have the synapses with the dendrites, which are exodendritic synapses. You can have synapse with the axon, which is called exoexomic synapses. You can have the axon synapsing with the cell body, which is called exosomatic type of synapse. So synapses, in terms of the structure, they can be a synapse between axon and axon, which is the exoexomic. And if it is a synapse between axon and the dendrite, it is exodendritic, which is the most common one. And then you can have exosomatic, where the axon is synapsing with the cell body. So I'll stop here. I would not go into the histology of the cerebellum and cerebrum and peripheral nervous system, peripheral nerves. This is going to be done in the in your labs. Thank you very much. If you have any questions, I'm here to answer.
Myasthenia gravis cause muscle fatigue to prevent.
Yeah, so because myasthenia gravis, the antibodies are against the autoantibodies are similar to they bind to the acetylcholine receptor. They would continuously stimulate the muscle, and over the time they would be.
Doctor, are there any immune cells in the brain?
Uh, Immune cells in the brain, no. But if the blood-brain barrier is somehow disrupted because of inflammation and damage, then the immune cells can migrate there.
The function of astrocytes, the part where they cover the capillary.
Okay, so they have the food processes which are covering the outer surface of the capillaries. So that forms the blood-brain barrier. Okay, thank you.
Doctor, one more question. Then what cell does the presenting cell present in?
The microglia can present as during inflammation, can act as antigen presenting cells.